everyone maybe one more minute as we let people trickle in, and we can get started.
So we don't have to move yet?
Not yet. Last 30 seconds. Okay, can we start settling in and we can get started? Welcome. This session focuses on wastewater surveillance in high-income countries with a specific focus on the U.S. domestic context. It's a two-part session. For the first hour, we'll have a rapid-fire series of presentations highlighting key work in this space from each of our presenters and laying the groundwork for the second half. And in the second hour, we hope everyone comes back. We'll have a moderated discussion between our two panels. On one side, it's research innovations in wastewater surveillance, and the second is providing more of the programmatic perspectives from the implementation side. And the idea for this session is it really echoes some of the byline of the conference of where research meets practice. So to kick us off, I just wanted to take a few minutes to provide a bird's eye view of the state of the National Wastewater Surveillance System at this time. I'm Heather Reese. I'm the epidemiology lead for CDC's National Wastewater Surveillance System program. So the National Wastewater Surveillance System, or NWS, launched in September 2020 in response to the COVID-19 pandemic. It began with a pilot monitoring SARS-CoV-2 in 8 states, and as you can see, rapidly has grown in the last several years. It currently includes routine sampling across more than 1,500 sites in 50 states, 7 territories, and some tribal communities. This GIF, as you can see, is not just showing the sites where sampling occurs, but is population weighted. The network covers about 150 million people, or over 45% of the US population. And for context, about 80% of the US population is connected to municipal sewers. In addition to SARS-CoV-2, the national program is expanded to include routine testing for RSV, influenza A, B, Mpox, as well as some influenza A subtypes. And in some states and territories, along with private and academic partners, also perform testing for additional infectious disease targets beyond those that have been officially onboarded into this national platform. So all of this is possible through a network of partners coordinating across multiple sectors. As you can see at the very top there, we have the first part of that puzzle of wastewater utilities who provide samples and contextual metadata. The second, over on the right side of the screen, are the network of public health, private, commercial, and academic laboratories who not only perform the routine testing, but also really are pushing the field forward with assay development and optimization research. And then down on the bottom, we have our public health partners, so including those at state, local, territorial, and tribal health departments, as well as CDC on the federal side. NWS also supports 6 centers of excellence that are located in California, Colorado, Houston, New York State, North Carolina, and Wisconsin. And they are really helping push this field forward, both on the science side, as well as being able to translate this into actionable data. So with the rapid expansion of this national surveillance network in a matter of just a few years, routine testing across more than 100 laboratories for a variety of different gene targets, and evolving methods for wastewater data analytics methods as we continue to iterate on the most effective approaches to translate these rather complex environmental data into a more cohesive public health approach across surveillance systems. We're entering a crossroads where we're trying to transition a national platform into a sustainable, robust, and agile surveillance data system that plays well with all the other partners in the field. So helping us kind of track this path forward, Very recently, in the last several months, NASEM, the National Academy of Sciences, Engineering, and Medicine, just released their second part of a review on domestic community level wastewater surveillance for ID targets in the US. This report was lengthy. I highly recommend you go out and read it if you have an interest in the space and you haven't yet, but it really detailed both some of the technical constraints as well as some of the opportunities practically to improve wastewater surveillance implementation. The recommendations spanned the gamut from optimizing the sampling network. Remember, we grew rapidly during the height of the pandemic, improving data quality and comparability, strategically expanding the ID targets that are included in a routine national program, and improved analytic methods to increase interpretability and use of these data. As we move into the presentations, we're about to shift that gear now, and the moderated discussion. I really want us to focus on those main gaps and opportunities to further bridge that space between research and a sustainable implementation at a national scale for this program. So first, we're going to go through our research innovations presenters. So I'd like to welcome Amy Pickering up from UC Berkeley. Let's see if we can get
First innovation, finding my slides.
Absolutely.
On PC.
Let's translate to Zoom.
Good. Okay. Great. Hi, everyone. I'm Amy Pickering. I hope you all are not as tired as I am, so you can pay attention today. I'm going to talk about wastewater surveillance for antibiotic resistance because I think it's an area where there is room for a lot of innovation, and there also are a lot of gaps in the research. And so I'm just going to focus on that today. And we did have some CDC funding to do some work as well, and I'll mention that today, but Susie Kim in our group is going to be doing a verbal presentation on that project later today, so I'm not going to scoop her with those results right now. Okay, so how do I, there we go.
There's also a clicker black.
Okay, yeah, clicker, yeah. Okay, so why do we care about antibiotic resistance? We have 5 million deaths per year globally associated with bacterial antimicrobial resistance, and we're living right now in a very interconnected world. So I wanted to talk about a recent study that I think is really interesting. It was an international travel study with participants from the United States that traveled internationally to various countries, and this was a metagenomic study that looked at before and after, so the same participants, before they traveled and after they came back. They did metagenomic sequencing and found that travelers were picking up new strains of Enterobacteriaceae when they were traveling abroad, and also had an increased burden of antibiotic resistance in the gut when they returned to the US. 67% of travelers acquired new strains of E. coli during travel, and this is a graph showing the proportion of travelers acquiring AMR-associated genes, and you can see that it's definitely a lot of genes are increased when they return home. So antibiotic resistance can spread quite quickly around the globe, and so that's why we should care about it on the global level. Here in the US, we also have a large burden due to AMR infections. 2.8 million AMR infections occur each year, and 35,000 deaths. And so there's a real need for surveillance, both to know the trends of what we know is a problem, but also to detect emerging threats for antibiotic resistance. And so surveillance is really important because the WHO and the CDC are always working on priority lists for what should be developed in terms of new antibiotics. And so we really need to know what's happening at the population level to inform those priorities. So the kind of like the traditional approach for AMR surveillance is clinical isolates. And so that's collecting samples from patients in hospitals. And so that ends up being a bit biased to those that seek healthcare and also doesn't capture asymptomatic infections. We all know that AMR is an issue in terms of genes being exchanged between pathogenic and non-pathogenic bacteria. So it's not ideal to be missing all of the asymptomatic infections. You're also limited by the fact that you can only usually culture a few species of bacteria and do limited susceptibility testing to different antibiotics. And so wastewater, right? Wastewater could play an important role here by capturing AMR profiles in populations. It includes hospital waste, but also community waste. And I think it has a lot of potential to really understand trends over time. Okay, so super boring slide, looking at different methods of what you can use to look for AMR in wastewater. We have culture-based methods, so that's isolating strains of bacteria. The strengths of that is you're looking at viable organisms. You can also look at phenotypic resistance, and you can also do whole genome sequencing, so you really know what isolates you're dealing with. But of course, this is a huge time burden, and I think a lot of surveillance systems are moving away from culture-based methods now that we have all of the next generation sequencing methods available to us today. And so I think we really need to be thinking about moving towards culture-independent methods for wastewater surveillance for AMR. So then there's PCR. So the advantages of that are that it's very quantitative, it's not that expensive, we have to know what targets you're looking for, and you also can't link antibiotic resistance genes to their hosts or their genomic context. Third method, metagenomics, which I think a lot of us are going to be talking about today, where you sequence all the DNA in a sample, and then you can do assembly to look at ARGs in some genomic context, but sometimes that's hard with short read sequencing, especially with highly repetitive sequence that flank ARGs. And you also have issues with low coverage. So, it's not very sensitive and it's hard to quantify abundance of ARGs with these methods. Okay, so little plug for the project that we did with CDC funding. So this was in line with developing dPCR methods for a broader range of targets than what people have been focused on to date. So thinking about looking at viruses, but also bacterial targets, fungi and protozoa, and also ARGs. So this is what Susie's going to be presenting later today. What we did is a first phase where we really tried to optimize methods for this broad range of targets in terms of the wastewater preparation. So we tried to optimize liquid and solid methods. And then in a second phase, we did some real world sampling. Oh, 2 minutes left. Okay, we did some real world sampling where we compared liquid influent, primary settled solids, and centrifuge solids, which I think a lot of people might be interested in because it's not always easy to get primary settled solids. So, results at 2:45. Go see those. Okay. I just want to mention a recent collaboration we had with Wastewater Scan. So this was a really nice opportunity to collaborate with them to apply some of the dPCR assays that we developed with the CDC project, but also expand to some other clinically relevant targets. So this study involved 164 wastewater treatment plants across the US, and we did kind of a cross-section of the concentrations of these genes. And so we just got this data and we're looking at it right now. It's really interesting. There's a lot of geographic variation. And we're also looking at what factors affect concentrations of these targets. Okay, so now targeted sequencing, really exciting new area. I'm being told I only have 1 minute left. So this is our preprint on a really new targeted sequencing method that we've been working on. That actually exploits the flexible programming of CRISPR-Cas9. So you can combine CRISPR-Cas9 with RNA guides to target ARGs. And what's really cool about this method is we're pairing it up with long-read sequencing so that we can look at the genomic context of ARGs. And we've been working on piloting it with stool and environmental samples. We're really excited to apply it to wastewater. I don't have time to talk about these results, but what are the research gaps going forward? I mean, I think we really need to be thinking about like what are the bug drug combinations that the CDC and the WHO care about, and what methods can we develop to really like do good surveillance for those. And so I think that's why long read targeted sequencing has a role, oh, it says stop, has a role to play. And so I think, but what's another really interesting question that I hope we can talk about today is, you know, what are the methods that we need to track what we know is a problem, but what are also, can we integrate other methods to be able to also track emerging threats? Okay, I'm done.
Thanks, and we're gonna switch over again, rapid fire, and then I think we're gonna have time to dive into the meat of some of this in our second hour. So, and this one. Oh, it's showing up.
Yeah, sometimes it's like yeah, see, maybe that's, now does it change? Yeah, okay, I think we're good. All right. Woohoo.
Well, glad to be here. I think my presentation very much builds on the other Amy P's presentation. Our vision is one of metagenomics as essentially a wide screening agnostic tool that can look at all sorts of things. And that includes antibiotic resistance genes, as well as pathogens and mobile genetic elements and virulence genes. And so there's tons and tons of potential here. And so the goal of our CDC project was essentially to assess that potential and then to look at the workflow from sample collection to concentration to extraction of nucleic acids, et cetera, and try to get as many targets across as many domains as possible. And so specifically, we compared a tight filter of 0.22 microns versus a loose filter of 0.8 with the thought that, well, most everything's associated with solids anyway, and you can get more volume through, right? And then we also compared looking at the liquid influenced sewage versus the settled solids with the idea that, well, let's let the primary clarifier do the work for you of concentrating your sample. And then we looked at kits specifically optimized for DNA versus RNA, since now we're playing in the virus world. And then ideally a total nucleic acid kit, so you get it all in one shot. And then we benchmarked our metagenomics to ddPCR and then also started playing with some targeted probe enrichment of viruses. So just some really high level results. So for example, looking at BAN-A genes that were present in Enterococcus that we spiked in, we saw a little better recovery with the 0.22 micron filter, comparable recovery with a DNA kit versus a total nucleic acid kit. And then the solids are here at the bottom. So a little bit, yeah, I'd say 2 logs higher number if you look at the solids. And then here is an example of an RNA target, so norovirus. And so we learned that there's tons of norovirus in raw sewage, and it didn't really matter what we spiked in, but that's great. It's a natural surrogate. And here it didn't really matter what the filter size was. And again, the total nucleic acid kit worked great. So that is good news because that's one step we can streamline in the process. We did play around with the TWIST Comprehensive Viral Research Panel, which targets over 3,000 viruses, and then we found mainly just like astroviruses and things not very interesting. We zoomed in on a few that are a little more interesting. And basically what we saw was, again, the total nucleic acid kit recovered more types of viruses. And then we also noted that we think that when we looked at the settled solids, there were a lot of inhibitors in there. So that was a drawback if you look at settled solids instead of filtering the influent. One of our goals was to assess the quantitative capacity of metagenomics. So you'll meet a lot of folks who will tell you that, oh, metagenomics is not quantitative, period, right? Not true, and we have a publication that hopefully will come out soon. We got very positive comments from reviewers, and so we're calling it Q-Meta, and this was led by my former PhD student, Ben Davis, and so basically doing a calibration curve of synthetic standards and got a nice linear response, but also figuring out where is that limit of quantification. And we benchmarked it for 4 ARGs and found a nice linear relationship when they're measured with ddPCR. Now, the final piece of our CDC project is to develop a wonderful user-friendly pipeline that can take all this metagenomic data and detect pathogens at the strain level. So that's the dream, and that's important, right? Because E. coli is not E. coli. Some will kill you and some are your friends, right? And so we knew what strains we spiked in of E. coli, and we also looked at Enterococcus here. And so this is just showing that our computer science team was able to recover those genomes and distinguish at the strain level for the ones that we spiked in. This is the pipeline. We're calling it Path Explorer, and that publication is in the works, but basically we use sour mash to screen and see what pathogens we think might be there, and then zoom in on those, and then do guided assembly and read mapping and so on. And then just to give you a sense of vision, so I'm also passionate about this tool for antibiotic resistance. In particular, I think that's where metagenomics can really shine, because there's over 3,000 known antibiotic resistance genes and hundreds of pathogens of concern. And so we've done a lot of work where we just annotate ARGs, and it's great for that. This is showing in a 2 local sewer sheds actually antibiotic use. So this is not metagenomic data. This is data we were able to get from clinics on inpatient and outpatient antibiotic use in these 2 sewer sheds. So you'll see there is a lot more in this sewer shed. And then when we profile ARGs with time, we had a lot more unique ARGs in that sewer shed as well. So it's kind of a nice demonstration of how for antibiotic resistance we can start to link to what's going on in the community in antibiotic use and assessing the efficacy of maybe mitigation measures like limiting use of certain types of antibiotics. So pros and cons. It's not all a utopia with metagenomics, but there are lots of wonderful advantages. I think, as I said, it is quantitative. I meant to, oh, here's the detection limit. It is high though. Our calculation is 5,000 gene copies per milliliter. And the strain level detection is promising, and it can work in some cases, but I don't think it's gonna work in all cases, especially with the shotgun approach. So I do think that we are going to need some level of enrichment, and so it's going to be kind of a combination of shotgun and enrichment. And we really need some nice standard spikes. I know, Zymo sells some, but we could have some that are more fine-tuned for the pathogens that we're interested in, because that was a problem that we had, was just, we spent half our time even developing our cocktail. And this is a publication we have that's to encourage standardization of metagenomics in the field, so please take a look. And thank you to my co-PIs, Peter Vickersland and Li Ching Zhang, and our wet lab team, and our dry lab team, and our postdocs. So thank you.
All right, thank you. Okay. All right. So I'm Lauren Stadler, and I am going to build off these two great talks in this session and talk a little bit about how we have focused on expansions of disease targets in our wastewater surveillance system. So I am part of, just as a little bit of context, I am part of the Houston Wastewater Epidemiology System. We are one of the centers of excellence. I partner very closely with the Houston Health Department. And our system consists, we monitor the fourth largest city in the country. We have a unique sewer shed system where we have lots of wastewater treatment plants. But we also have a very comprehensive facility level surveillance system where we monitor at schools, nursing homes, homeless shelters, and the jail. But this kind of unique system allows us to really think about target expansion, both in the context of surveillance at the citywide level, but also target expansion at the context of an individual facility. So what I'll focus on today is some of the research focus that we have been working on, really thinking about how do we expand the number of targets that we're including in our routine surveillance. And so the kind of the overview of how we do this as a system is we have a group or a system for identifying and prioritizing new targets. We have an advisory board. We use them to help us establish our sampling strategy. So not just what to look for, but where to look for it. And then I'm kind of the lead of the lab team. So we get to work at designing and testing those assays and implementing them to collect some baseline data. And in the meantime, the health department collaborators are thinking about, once they get this information, what are they going to do with it? Who's going to get that information? How are they going to communicate it? What's the action that's going to be taken from it? So we have a plan in place before we start looking for things. And then we integrate that, try to establish the thresholds for actions, and some of it gets integrated into routine surveillance, but a lot of it doesn't. We generate data, it's not that actionable, it's not worth the resources, and we don't include it. Because I only have, probably 5 more minutes, I will say, I'm really focused on how we design and test new assays, and that's kind of a lot of the research that we do. So for context, we kind of have 2 major pipelines that we're working with in our lab. One is trying to figure out how much of X disease target is there in the wastewater, and then a second is, we do a lot of targeted sequencing. So the 2 previous presenters talked a little bit about this, but more on the metagenomic sequencing. We do much more of targeted sequencing, and the vast majority of, for example, SARS-CoV-2 genome sequencing that we're getting out of wastewater is obtained through targeted amplicon sequencing, where we're just amplifying the genomes of interest out of wastewater. And that's to identify circulating variants. So the research areas that we focus on, and I'll just touch on them because I don't have time to go into the results. One, and this is part of our CDC project, was to really just kind of compare concentration methods to assess them in terms of sensitivity and cost as we expand from just a handful of viruses to a suite of pathogens that include viruses, bacteria, fungal targets. Second is trying to develop computational tools that enable faster design of tiled amplicon sequencing primers for new pathogens and for existing pathogens. And then the third is kind of an extension of that, an application of that, where we use this computational tool to design primers to target a suite of pathogens in a single reaction, and then thinking about how we make that quantitative. So just a little more detail on each of those. I think I have 1 slide on each. With not a whole lot of results, but first, we're thinking about concentration, which is really, I think, kind of at least in our lab, it's always been kind of the most critical step in terms of our recovery and of our overall detection of all of the pathogens that we care about. And so we've compared a handful of different methods that are commonly used for kind of a longer list of targets. And for each of these targets, we're concentrating them in these 4 different methods in this head-to-head. And then we're comparing the sensitivity, and then we're also doing an analysis of kind of the equipment, labor, and time costs for all of these. And kind of the results helped us decide the method that we wanted to use. And when it came down to it, it's really target specific, which of course, when we knew that going into it. And so there's going to be trade-offs. One thing I will say, there's a lot of move towards magnetic bead capture technologies, which turned out to work quite well, but they are expensive. I don't know how many health departments who tried to use these have done the math on this. It is really expensive. And so that's really one of the barriers, I think, for pushing down. And as people standardize towards things, we have to think about how's that going to impact the overall resources available to our programs. Second, in collaboration with Todd Trigan, we have been focused on developing computational tools for designing tiled primers for amplicon sequencing of pathogens out of wastewater. So we developed this pipeline called Olivar. We have a publication on it if you want to look at it. We actually have a website where you can go and use it and try it out. And the idea here is that you could input your pathogen genome of interest and it would output a set of primers that are largely, it's automated in terms of developing these primers that will work to amplify your target genome out. And so we actually used it on SARS-CoV-2 and we compared it kind of to the state of the art, which is Arctic primers, which a lot of groups are using. And we showed that our nearly completely automated design primers worked as well, in some cases a little bit better, on a wastewater sample for amplifying SARS-CoV-2. So now we're using Olivar to design primers for other targets. We're also using it, this is kind of the extension of it, to design primers that allow us to target multiple pathogens in a single reaction. So instead of trying to maybe extract to amplify the entire genome of every pathogen, we're kind of saying, what are the specific areas of the genome that are most informative, and how do we design primers for many of those genomes in a single wastewater sample and multiplex it in a single reaction? And so we're developing computational tools for identifying these signature regions of these pathogens, and then to do the automated primer design that kind of balances, primer dimers, avoiding SNPs, and making sure you target regions that are informative for variant analysis. And then finally, we're also thinking about how do we make this quantitative? Because again, one of the big challenges is as you start doing targeted amplicon sequences, we're going to lose the quantitativeness of this, because this could be really powerful if you could multiplex hundreds of pathogens in a single reaction. But a lot of times, if we still have to do the quantitative ddPCR, we're not necessarily going to have as many potential cost savings. So the way we're doing this is we're using something called unique molecular identifiers, which are basically kind of a trick for basically figuring out how many initial molecules of your target were present in the sample. And then you can use that information to get absolute quantitation of those targets in this multiplex reaction. So this is something that was started by this CDC BAA project, and we're still actively working on it, getting promising results showing that this can be quantitative for many pathogens in a single reaction. So I'll just back out and say these are kind of the three major areas. And again, this is all in the context of thinking about how are we going to expand to target, not just 3 respiratory pathogens or 10 pathogens, but maybe even hundreds of potential pathogens. How do we rapidly design primers so that we can implement this for the next emerging pathogen and integrate it into our routine surveillance? And so with that, I will stop and just thank my group and my collaborators who did this.
Thanks so much, Amy Pickering, Amy Pruden, and Lauren Stadler. We're going to shift gears a bit to our programmatic side. So we have 4 presenters joining us, all from different health departments that are National Wastewater Surveillance System partners. And to start us off, we're going to move to one of our virtual presenters, Carrie. Can you, perfect.
Can you hear me?
Yes, and let me just expand your screen so that we're not looking at a little thumbnail.
Yeah. Can you see my slides?
I can, I'm trying to move them over.
Awesome.
Okay, you're good to go, Carrie.
All right. Hi, everyone. This is going to be a little different than the previous talks. So today I'm going to talk a bit, I'll start by introducing myself. So my name is Carrie Regan. I'm the wastewater surveillance epidemiologist at the Utah Department of Health and Human Services. And today I'm going to talk a bit about the Utah Wastewater Surveillance System and some of the things that we've been working on recently and some of the things that we're thinking about. So let me start by just introducing you to the history of the program. So back in March of 2020 through May of 2020, we started with approximately 20 sites, and this serviced about 40% of the Utah's population. Sampling was done once per week at 3 different laboratories at universities. These were unified methods, checked frequently to make sure that they were providing similar methods. We then expanded to later in the summer to 47 sites, still sampling once per week, and we were one of the initial 8 members of CDC's news. However, during that time, it became clear that the data was not timely enough. We needed sampling twice per week. However, the program could not afford sampling twice per week at 47 sites. And also some of our more rural sites, people were not able to provide those samples twice per week. So we had to drop some of our sites, and you can see that the brown squares are sites that we ended up ultimately deciding to drop. And that brings us to what we look like today. So today we have 35 community level facilities servicing about 88% of Utah's population. Utah is a little bit different in that 92% of the population is sewered. I know that's a very unusual stat for a state. So we're able to get quite a good look at the state's population. We still do sampling. We now do sampling twice per week since January of 2021, collecting mostly 24-hour composites. And lab analyses are all done. They were moved to Utah Public Health Laboratory, where we do ddPCR and whole genome sequencing. And just a quick look at what our data would look like to the public, because we provide information through dashboards and websites to the public. This is kind of what they would see. So they would see the sewer shed that they're interested in, a categorization of the concentration and trends. And so you can see here we have wastewater at the top, case data at the bottom, and kind of some information about historical concentrations for the public. And so one thing that we've been thinking a lot about since the guidance about right sizing is kind of how do we pick sites and the sustainability of our program as a whole. Will we be able to keep 35 sites forever? And I'm not sure. But some of the factors that we are keeping in mind when selecting sites permanently are things like local health department interest. So that's in the case of Heber City and Midway, 2 sites that we removed initially, but brought back because of Wasatch County being interested in the data, using the data for public health action in their jurisdiction. We also try to select sites that are 24-hour composites. However, there are a few exceptions to that in areas that we really feel are needed throughout our surveillance system. And then, of course, when selecting sites, we need the data to be useful. Preferably, if we were going to bring on a new site, we would want an area without good coverage. So this would likely not be Salt Lake City or the Wasatch Front. Maybe the site would provide some data addressing health gaps, and would be of interest at the federal, state, LHD, or community level. And this is kind of what I've been thinking about a lot is expanding to new targets. So we are currently in the process, and we've expanded to a few new targets recently to thinking about non-SARS-CoV-2 targets. And we have to think about this from a funding standpoint. How much money do we have to do this? How sustainable are these different targets? And we're thinking a lot about which targets to select and where to do them. So ideally, the targets that we're selecting, we would have a plan outlined for them already, working with other epidemiologists throughout the Department of Health or local health departments. The targets that we select are hopefully going to be useful for the community. Obviously, the detection is feasible in wastewater. We collect liquid influent, so we don't want to be going for things that are solids. And we've recently moved to the new multi-target panel developed by CDC. And so this is including targets that are influenza A and B. We're doing H5 subtyping, RSV A and B, but we're also expanding to CRS. And although that is not something that's done everywhere across across news, is something that is of particular interest in the state of Utah due to some Candida auris outbreaks in our neighboring state, Nevada. And so we're hoping that this provides a lot of interesting information. We're also looking to expand some of our sites for MPOX. That will likely not be every single site. And so we're doing a lot of thinking about what is sustainable, what is doable for our surveillance system, and how exactly do we move to these picking sites and new targets. Some other future plans across our surveillance system is data automation. That's always helpful. That always makes things more sustainable. And then integrating new types of data. So here I have a graph just showing integration with emergency department visits. So showing wastewater data and emergency department visits, kind of seeing if we're seeing similar trends there. And then we're also looking to do further integration with some modeling at the University of Utah, neighboring, it's a university in Salt Lake City. And they are implementing some wastewater into their forecasting and nowcasting models to be able to do some hospitalization counts. So this is all very interesting, great things that are happening here within the state of Utah, but also with keeping in mind some of the sustainability efforts that we're trying to think through. So these are some of our ideas and some of where we're at right now. And yeah, if you have any questions, please let us know. Thank you.
Let's hold those questions till the second hour when we return. Thank you so much, Carrie. Dan, can you present your slides? Perfect, we can see them. The floor is yours, Dan.
All right. Hi, everyone. Welcome. Happy Friday. I'm Dan Lang. I am the Deputy Director of the Center for Environmental Health here in the New York State Health Department. And I always say if you're involved in the wastewater program in New York, you can blame me because I kind of got it started during COVID and it just ballooned into a pretty robust program with lots of people contributing to all of this that I'm about to present, including our university partners, Syracuse University in the central part of upstate New York, have been instrumental in putting a lot of all of this together with us and now expanding even further into additional targets, just like Carrie had mentioned in Utah. Let me see if I am advancing my slides. Not advancing my slides. All right. Here we go. All right. So we had a fun time at this. We got together and tried, everyone likes to come up with a mission statement. So we did so. And also in the process of doing that, we put together a little cartoon that's right on our website that describes what the wastewater surveillance program is and what it does and how we get information out to the public. So that's kind of when you go onto the New York Wastewater Surveillance page, you come to that little cartoon. But I know I don't have a whole lot of time to do this, but we have a neat little cartoon. Okay, so where are we in our testing program? So we have 62 counties in New York State, including New York City counties and the boroughs. And so we have 205 wastewater treatment plants participating in the network in every county in the state of New York. So that covers more than 15 million people. And I think there's roughly 85% of the population that is on sewers. And we're covering about 80 to 82% of all of the sewered population in New York State. So it's quite the big program. This kind of gives you a little breakdown of what percentage of the population in each county that we're covering in our wastewater network. And we expanded that very rapidly. I mean, similar to the news national program overall, this program in New York expanded rapidly, and I'll go into a little bit of how we did that. So back in March of 2020, I call that kind of our visionary period of time when we were just discovering that we could detect the RNA of SARS-CoV-2 in wastewater, and then embarked on a bunch of different pilot projects Our pilot project that we did with Syracuse University in August of 2020 was around 4 municipalities throughout the state: Buffalo, Rochester, Syracuse, and Newburgh, New York. And we did upstream sampling in that pilot. So it was many samples and collecting samples from manholes and automatic samplers. And we soon realized that that was going to be way too expensive to expand to the entire state of New York. So we went back to what's the traditional way of collecting the samples, mostly at wastewater treatment plants. Joined News in 2021 and then began in early 2022 with a rapid expansion from that 12 sites to 200 sites. And really, one of the lessons we learned there is just, it was a day-to-day phone calls and emails and just building relationships with wastewater treatment plant operators and local municipalities and our local health departments. So it was really a grassroots effort that got us to that point. And I think it really helps for the long term too, because now we have these relationships. We provide weekly memos of all of the results from the wastewater program, customized weekly memos to our local health departments and wastewater treatment plant partners. And so they feel like they're getting something out of their volunteer work to collect these samples, and they feel like they are part of the network. That has helped to maintain our 200 sites participating in the program, even post-COVID. And then I'll talk a little bit more about polio before, but you probably all know that we had a paralytic case of polio in July of 2022 here in New York, in Rockland County, which is down in the close to New York City, down in the metro area of New York City. And that was really something that really got the attention, I think, of many people, at least in government, of how useful wastewater surveillance can be and how rapidly it can be used to assess an outbreak. And then we added more capabilities to the network. We had a sequencing consortium that was doing clinical whole genome sequencing of SARS-CoV-2. We were able to adapt that to wastewater. And now we're working on other innovations, adding flu, RSV. We've done some pilots on Hep A and norovirus. We were also in the middle of assessing our Eclipse study. So we had a pretty big event last summer. Eclipse, as many of you probably saw, came through New York or the states, but went right through some really high populated areas in New York State. And we wanted to assess whether human movement during that event could be picked up in our surveillance system. So we're still evaluating the data on that. So just some of the achievements, I guess, or highlights of the program in New York. One of the things we always got asked, especially internally by our commissioner and others, is, all right, great, you got all this data, but how is it being used? Who's using it? Who's actually even looking at it? And so we put together, this is just a small sampling of the amount of press, and most of this was started by the locals, right? So local mayors and town supervisors and others who were really on the front lines of providing information to their constituents would take a look at our wastewater levels, and get the news, get it into the news, put out press releases about whether levels were increasing or decreasing. And so we have a lot of use cases, I guess, of getting information out to the general public about wastewater levels and how they relate to cases and hospitalizations. As I mentioned before, the sequencing consortium. So we have 5 laboratories throughout New York State that work on clinical specimens, whole genome sequencing. And then we turned that whole consortium to wastewater. And so now every positive sample from all of those 200 sites every week goes to our consortium laboratories. And so we have a very robust understanding of the different lineages of SARS-CoV-2 that are in our communities. And so you can go look at a New York statewide map, and you can also zoom into each county to see what lineage or variant is circulating in that community. And then polio. So if anybody, I think one of the best use cases really is poliovirus. The detection of the case was first. So there was some AFM information and surveillance out during that time. And, a doctor had a patient that had some symptoms similar and collected clinical samples.
Dan,
And,
sorry to interrupt. So sorry. Could you wrap it up? You're at time. Thanks so
okay,
much.
very good. I couldn't see whether. So anyway, polio, good use case scenario, retrospective sampling as well as ongoing. We're still doing, even through 2024, have been continuing poliovirus testing, although we have not detected polio, thank goodness, since 2023. All right, that's it. I'll wrap up.
Thanks so much, Dan.
I'll speak extremely quickly so that no one understands
That's what I was going.
Yeah, haiku style.
Perfect.
Thank you, Molly. Hi, so my name is Molly. I am with the North Carolina Department of Health and Human Services. I'm a genomic epidemiologist. I work with our communicable disease branch, so mostly with human clinical data. So the goal of this presentation is to put some of the wastewater data that we have in North Carolina in a broader context of some of our other surveillance systems, particularly our clinical surveillance system for SARS-CoV-2. So for our clinical data, we began sequencing SARS-CoV-2 regularly sequencing SARS-CoV-2 back in January of 2021. We established an academic partnership across the state with several of our universities in order to do some of this, including UNC, so thank you, UNC. We also import data from private labs such as LabCorp, which is done through a contract with CDC. And we use these data for a variety of purposes, but our main one, I would say, is to update our weekly variant dashboard on our state COVID-19 website. So it's very similar to many of the variant dashboards that I'm sure you've seen at this point. We just have the different variant prevalences. So this shows our sequencing as well as our reported case data over the course of the pandemic. You can see that we had all of the spikes that everyone's very familiar with. So the case data is in the blue bars, and then our sequencing data are the red dots. And you can see that basically over the last year, our sequencing has been a bit of a flat line. And if you zoom into the data from 2023, you can see a pretty steep drop in our sequencing beginning in January of 2023. I also marked off the end of the federal public health emergency, which was in May of 2023. Not only did the federal emergency end, but here in North Carolina, that meant that COVID-19 was no longer a reportable condition. And also importantly, the funding for that academic partnership that I mentioned ended. So we expected to see a huge drop in sequencing around last summer. We were a little bit surprised to see that it actually started before then, really in the winter of 2023, but you can see that over the past year, or this is from last year, or sorry, this is from 2023, but over that past summer, even when we saw an increase in cases in the summer wave, our sequencing still stayed pretty low. And these numbers are a little bit, this is through the end of 2023, but since the end of the public health emergency, we've only been sequencing about 34 samples a week. Some of our weekly updates on our website have had fewer than 10 sequences, so it's pretty low. And this is not only happening in North Carolina, this is from the CDC's COVID data tracker. This is just from last week. And you can see that they have a nowcast model where they estimate prevalences for each region. And in order to run the model, there have to be at least 300 sequences in order to have stable estimates. And you can see that last week, only 3 of the 10 regions had enough sequences. And North Carolina, our region, did not meet that threshold. So we've seen this steep drop in sequencing both in North Carolina and nationally. It really limits our ability to have stable estimates for the various prevalences. It also limits our ability to detect new variants, emerging variants. And so it's really made us think about the role of our passive clinical sequencing as part of a larger multifaceted surveillance system. So, not surprisingly, this is where wastewater sequencing comes in. So as several folks have shown, particularly for SARS-CoV-2, what we see in wastewater is really similar to what we see in our clinical sequencing. So it's not quite the same, but you can see similar patterns in our clinical samples and in our wastewater samples. So this is really exciting. And so it's really forced us to think about, particularly for SARS-CoV-2, but I'll also talk about some future targets. Like what is the role of sequencing for SARS-CoV-2 at this phase of the pandemic? Is it for variant or for to update our vaccines? Is it to have a general understanding of prevalence? Is it to detect whatever the new emerging variant might be? How should we be thinking about our passive clinical surveillance versus active surveillance? How should we be incorporating wastewater or other surveillance systems? And then for all of these, how do we create a sampling scheme that allows us to actually answer these questions? We are certainly thinking about that in North Carolina. I know that other states and CDC and academic partners are thinking about this as well. So this is all to say that we see wastewater really as one piece of an overall comprehensive genomic surveillance strategy. So wastewater provides a community level picture of transmission. Our passive clinical surveillance tells us what's happening clinically. It might help us inform therapeutics or vaccines. We are not specifically sequencing outbreaks. We know that there are some jurisdictions that do, but sequencing from outbreaks can give us some insight into high-risk populations or transmission dynamics. If you can get really high-level sequencing from an entire outbreak, that can really tell you a lot. And then hospital-based surveillance can also help inform if there's a particularly virulent strain, or if we were to see a divergence between our passive clinical sampling and our hospital-based sampling, that would be very informative. We're not currently doing that either, but something else that we're thinking about. Which also brings me to new targets. So many folks have talked about expanding our wastewater surveillance programs. These are just some of the targets we've talked about. H5N1 is already happening. It's up on the CDC database or tracker, I believe. But we've talked about measles as well, and then of course, as we've heard today, antimicrobial resistance genes. But as a molecular epidemiologist, I'm really focused specifically on the role of sequencing and genotyping, and so all of this has generated even more questions, which is how will sequencing change our understanding of disease transmission? Will it change our public health action? Do we even need to sequence? There's a lot of excitement around next generation sequencing and metagenomics. I'm not against it, but there's costs to it. And so could we use other PCR-based genotyping methods? And then in general, how will sequencing from wastewater fit into that broader comprehensive picture? So that was really fast, but thank you.
Thanks so much, Molly. We have one more quick presentation from our last health department partner, but I know that we're at time for this first session. We can take a very quick break, I think, and then we'll start our second session with that last presentation and then move into a discussion and bring back everyone who was here presenting. So let's take a few minutes break.
Should have gone up earlier.
Oh.
Looks like you got it all. Looks like you got it all. That's how we
I hope we got it all.
All right.
Ariel, where's your presentation?
So you're doing the
Because you need like enough.
At least submit it. And then.
Have you, could you have like 2 library?
Yeah,
projects? Yeah, that's what you're doing.
I mean, it's like we're just doing the brute force of it. That's what I feel like, but it's not like good. It's like,
Yeah.
I could fractionate things all day, you know? It's just, it's not ideal.
Yeah.
I like that last talk so much because this is the conversation we have with our health department partners over and over and over. Like they don't care about the sequencing data. Like they literally don't even look at it. Like if I don't send it one week, nobody tells me. We didn't get it.
So at CDC, that's a problem too, right? And I think if we talk to Dan Horn, it's like, oh my goodness,
Still clear when
why can we have this as, in my mind, it's just that's where the greatest is that experience. Not doing anything with data.
Yeah. We need a better.
So we need like,
I think it's a
we need
combination.
how to interpret.
We need to have interpreters on a national level in mortality data, because I think what we're seeing is actually coming into CDC is you're like, part of the whole point of this is it's not just increasing geographic coverage relative to clinical sequencing, providing a less biased sample, but it's also the timeliness factor. So every time we get like, well, usually it's a month old, the only thing that actually came in within the last couple of weeks is like a single state.
Yeah.
What can we do with, exactly, and so it's,
But it's hard to, it's like a chicken
No, it's
egg
a-
problem, because like people are not standing up for sequencing because they're not seeing use of it, so it's getting deprioritized, so it's getting further delayed.
no, agreed. I think we need to figure out, what I'm hopeful is that Dan has the bandwidth to lean in on the side to increase the collaboration and like pull this together, because I feel like some of those
I know, he's
there and we're just not. So I feel like we have the ability to shine the visibility on it, which I think will help with this.
I was thinking, last time I talked to him, he was like, you know, we need to use SNAP-22 for like test kits for the next thing. So like you need to have the infrastructure in place so that like whatever is next, you have this here already.
My concern with using, so yes, and not my area of expertise, so everything I need to get quickly out of my depth here, but I feel like just from like pragmatically, even the clinical genomic surveillance for SARS at CDC is really effectively beyond this. What is your, what are the proportions? Like, I don't know how actionable that is. There's been minimal efforts related to taking it to something like, I don't think, like, once we got out of the intra-agency space where it was very clearly driving questions about therapeutics, like recommendations, I'm not saying, as soon as we stepped away from that practical piece and the public health emergency ended, so there wasn't that weekly touch point across the intra-agency, like, I don't think it's usable. I don't think that having variant proportions is usable in the way it's currently displayed. So I almost feel like shifting gears to something else where people aren't used to seeing data in a certain way, and it's hard to pull it and like have that new might actually be a better inroads if we could say, well, look at this. But no, I understand there's a lot more data there, there's already a better system set up for us.
I'm just trying to, I'm trying to make the case also in our health department. They're so actually like they're so
Well, that's what I think is that we were so used to this immediate tie-in to.
I'm still.
I think that's the issue.
Do they use it for vaccines or not? I have no idea.
I just think it's like, it's the best.
I do think
Or like when a new. I feel like when a new.
Boosters should be pushed. I've also been used to, like, sequencing has helped people realize that their diagnostic assays are wrong.
Yes.
So like there's a bunch of cases where people have done that for like a bunch of different viruses, which is very powerful, right? Maybe that's when you have to do like it. I don't know.
I think that's a difference. Then you're like, well, I don't need to be as time. I need to just be
He's very annoying.
I did, but it's okay.
I would say they were.
He's very much like.
It's funny.
Thing to you, I wanted to bring up this whole thing of like this rapidly
emerging issue. Yes, we have this right. How do we transition quickly and punt it to the Texan here with H5? And talk about this.
but I feel like he's having that in like all of these different but like no, but like other different groups that are trying
Yeah, they're trying to
and then everyone's trying to figure out what it even means in our
I think that's a really nice.
I like that we took the casual approach
Situation that life is like
I know, I'm like, okay.
parts. If we're developing it at the same time.
I would have not done it like that.
Still in the space of like,
This makes it great, you know, like,
everybody, all hands on deck.
exactly, and then you go to the implementers and they're like, whoa.
Yeah, and then
CDC
that's forcing us to do something like that. I feel like it's exactly, they're like, what is this thing? And we don't want to look for it because.
I know.
Trying to pull it together here.
good work. I want to I have like samples and like if you can just like let's collaborate on some of these topics because like but like if you're up for it, like you know we can figure out like because I just was like wait, we need to look at the paper.
Oliver? Yeah.
No, I
That was
took
all
the
on start. I
immediately gave up my office.
But no, I
and was
want to
like,
like,
yeah, that's
yeah, I want to expand it because like I'm doing like a little I have some and like our collaborators like my machine died, do you have a new machine? He's like, no, I don't have an extra. Yes, and so like if there's a way like that we can
Yeah.
and like I mean we're thinking about this, but we don't have that platform that you're doing. That's what I was
And the guy who did the post. I know, I'm trying to get him as a postdoc, but I'm kind of like, he's actually in child training, he usually works like a child lab, and I guess you're advisor with a postdoc, because he's design like, you know, I got some child primers for these, X number of pathogens. He was doing things about this, really for taking it over to your lab so they can do it, and then do the insulin supplementation side to renal component of that, so I can have it, how do you make sure it works, especially if you're really
You already pulled them up, thank you.
I was like, there's such a hit right. I mean, we're at 11:10. I don't know when this.
Yeah, when it technically, I feel like maybe technically started. But maybe I'm wrong. I looked at it, you're right.
10 to 11:15.
But it's exciting. Think about
I think 11 or like 10 minutes after 11, so we've done a 10-minute break.
Thanks, everybody. We can start taking seats and we can do our
last quick presentation, and then I invite the panelists who are in person to come up and sit at the table after.
You want to be a company.
Okay. Thank you, everyone. If we can quiet down, and we'll restart. Wonderful. Okay, thank you so much. We have one last presenter from the North Carolina Department of Health.
Thanks, Heather. Thank you all for coming back. I'm glad that you did, because we're going to enter story time now instead of specifics around research and findings. We're going to talk a little in anecdotes and narratives today. So I'm going to talk a little bit about public health action and response. And I think Lauren Sadler's timeline that she showed earlier around, lab method development, but then also the work that was happening in the health department side of communications and what we're going to do with these results is happening in parallel. I really loved that. And so I'm going to talk about that other part of the box here about, how we're developing some of these things. So first, I really wanted to brag and start us off by saying that this year we were awarded a Center of Excellence here in North Carolina, and I just really want to attribute that a lot to our UNC partners and UNC system research partners across the state. Angela Harris is back there with NC State, and our partnership with all of these system researchers, I think, really made us have a really strong application for being able to be a Center of Excellence here in North Carolina. And we were also one of the first 8 states that were funded for CDC News. So just really continue to kind of partner well with our research and health department partners and try and kind of close that gap between the two. So the first story I'm going to start with is something that we were doing in collaboration with Wastewater Scan, and I think they've been mentioned a couple times here today, and I think folks are familiar. I see shaking of heads with how many targets they're able to do across the country in partnership with them. We have 3 sites here in North Carolina, and we really started seeing something after about 6 months of non-detects with hepatitis A. At all of our sites. At one of our sites, we then started to pick up seeing consistent hepatitis A detections. So we were really like, something's going on here. Wastewater Scan site is something that we check almost every day, as well as like our local partners are very aware of it, and they are also taking a look. So in terms of timely, actionable data, it's been really useful for us. This hepatitis A situation was something we were, very concerned after seeing this period of non-detects and kind of establishing a baseline of hepatitis A at some of these sites. When we started seeing detections, it felt like something we needed to respond to as a health department. So I'm one to not bury the lead on results. So I wanted to really talk about this hepatitis A situation from one of the major findings that we had, which was that wastewater detections began 12 days before symptom onset for 2 clinical cases that were identified, which I thought was a very fascinating finding of this overall investigation. Hepatitis A is a pretty newer target, I would say, in wastewater surveillance, comparably to like how much baseline data we have for COVID-19. So we really didn't know at first when we started seeing these consistent detections if the pattern, what do we know about shedding, right? And for this particular pathogen, and do we know if this represents one case, or am I looking at an outbreak of hepatitis A that we're going to have to implement multiple partnerships and look at some of these high-risk settings, which we know to be problems, right, for hepatitis A. And we also, really didn't know much at the beginning about comparable levels of some of these concentrations. So we started seeing increases, but we weren't sure if those increases were significant or actionable, right? So one of the things that we really wanted to do when we got the first detection on March 16th, we, kind of had some conversations about it, and we, like others have mentioned here today, we had a plan in place for all of these targets that Wastewater Scan does, but also obviously hepatitis A for what we were going to do and what point was actionable for us. And I'll show you guys that in a minute. So using both wastewater and clinical and traditional surveillance, we were really able to align these two peaks that we saw for hepatitis A with individual case patients. So we got, after our third detection, we ended up being able to do some notifications, which I'll show you in a few minutes here. But patient X was not identified. The first patient wasn't identified until April 5th. So March 16th was when we had our first detection. So that really kind of shows you the lag in clinical surveillance for being able to detect these cases and the potential for wastewater to be able to give you an indication of something happening in the wastewater sooner. And the second peak there was the same situation where we had the suspected infectious period starting, and basically the next day was the first wastewater detection that we saw, which I thought was just a very fascinating part of this whole story. So as promised, we had these plans in place before we started doing any sort of testing. And we're not doing this at the health department, but we really consider the wastewater scan sites part of our overall network. And if we're seeing things in the wastewater scan data, we really feel that we have an obligation as the health department to be able to respond to anything that we're seeing. So it forced us to look at some of these new pathogens, and I think was demonstrated a really great relationship between research and health departments so that we could kind of pressure test some of these targets and see if we could be able to pull them into our routine surveillance. So this is just showing kind of like a draft public health action, what we call a public health action decision tree, where we, after 3 concurrent detections, we notified our local health departments. We had a conversation with them about potential sources for what we were seeing for this hepatitis A signal. We ended up doing some additional outreach to hospitals, which was all outlined in this public health decision tree that we put together before we even saw these detections. So really what we did here in this investigation was we just tried to pressure test that public health action decision-making tree and see how well it worked. And I do think it was very successful. We were able to notify the health department or notify the hospitals and also look into syndromic surveillance to see if there was any cases that were identified using those. We also talked a lot with our hospital partners about high-risk populations and did some outreach on April 19th when we started seeing those additional, we had a period of non-detects and then we started seeing this go back up. So we thought something was still going on and we potentially had community transmission. So we talked about, we did some outreach on Facebook and we did some outreach to some high-risk populations that we've been dealing with hepatitis A outbreaks in those communities before. So we had those kind of pre-established relationships set up so that we could do outreach. And I will talk briefly about another story, which is, of course, the H5 situation. I know I'm a little bit over time, but I'm gonna just mention we're doing H5 testing in wastewater, and this again was kind of born out of a collaboration with Wastewater Scan and that close relationship between research and partnership and health departments. So this is our 3rd season of conducting influenza A testing in wastewater at select sites. And what we saw this season was after about 4 weeks of non-detects, after the kind of clinical season started, we started seeing things uptick in influenza A. And of course, that was around the time we were concerned about H5. We were concerned, CDC was asking questions to us about potential animal outputs or inputs that could be contributing to the system. There was just a lot of question marks. So we worked with Wastewater Scan around the time that the first case patient, I think, was identified in Texas. To be another site for this inaugural paper, I think, that demonstrated being able to subtype H5 in wastewater. And what we saw in one of our sites that was participating in North Carolina is we saw the majority of the signal that we received for influenza A was actually H5. And that was further, after some investigation, loosely linked to a milk processing facility that we were seeing in North Carolina that was contributing to the sewer shed. Those no point, specific testing was done, but we think that there's pretty strong evidence that that was the contributing factor we were seeing there. Again, that just really demonstrates the utility of working with some of these research partners and then thinking about how to translate that into public health action for health departments. We are now doing influenza A testing at all of our sites across the state. We are developing in partnership with our university partner, which is UNC Chapel Hill, Dr. Rachel Noble down at the Institute for Marine Sciences, a subtyping panel, which we've started implementing for being able to test for not just H5, but also H3, H1, and really just being able to know if we're seeing an increase in influenza A, we need to be able to not just say it's not H5, but what else are we potentially seeing in the wastewater. And this is, again, this draft of a public health decision-making tree that we're really trying to pressure test. So we always love to hear feedback from folks about some of these. In public health, I like to say perfection is the enemy of good. And I think we often operate with impartial information. So really trying to come up with these decision-making trees and not just talk about anecdotally how we could do this, but really try and make decisions and then get feedback from folks on how well those decisions worked and evaluate is really part of, I think, the whole public health process and thinking about how to implement some of these new pathogens moving forward. So I'll stop there. Thanks for letting me take a few minutes.
Thanks so much. And can I invite all of our presenters to come up? We're hoping this can be a discussion with the audience, with our presenters up here to tackle some of these main pieces. Dan and Carrie, please just turn off your mic when you'd like to speak or indicate. I know it can be a little tricky when you're trying to kind of go back and forth between the both. I'm happy to kick us off with the first topic for discussion, and then we'll refer to the audience. But Ariel, starting from where you let us off, I feel like at a national surveillance system, we have a plan of what core targets and kind of often for endemic pathogens we're interested in implementing. We have this solid plan, and then first Mpox gets thrown at us, and then HPAI gets thrown at us. What are those different pieces that we need to be thinking about from the rapid assay development and testing and piloting in wastewater? And then Lauren, nope, you're starting. From that piece, and then also thinking about what do we do with this data? How do we make the determination of utility? And then eventually, how do we think about something that can actually be scaled and sustainable in a program? So.
Yeah, so I feel like, I don't know if this is working or not. There we go. I don't know if I just, can you hear me? So I think H5 is a great example of this, where we went through this in real time and thought about, okay, how do we rapidly respond to an emerging threat, and how do we kind of all hands on deck, and what do we learn from that experience? And so I'll just talk about our experience in Houston and in Texas, where there was a really large active outbreak, and actually our partners at the Texas, TEFI, I don't know if the acronyms are, the Texas State basically monitoring system. We're doing some more agnostic screening where they're using probe-based enrichment, which actually was very powerful in this case, because if you don't know what you're looking for, this is a way to discover things that are kind of closely related to things that might be of interest. And so they were able to detect the presence of H5N1 very early in Texas wastewater samples, and that allowed us to kind of scale pretty quickly to do more kind of monitoring at many more sites to see, how is this widespread? Where are we seeing, and what are the possible sources of this? And I think we learned a lot. One was that rapid data sharing across, not just within Texas, but nationally and even globally, is so critical for fast response. So a lot of our sensitive detection techniques that involve designing primers require that we have some knowledge of what we're looking for. And a lot of times that means whoever's detecting it first, if they're able to get any information and share that, enables everybody to rapidly respond. And so we really learned a lot from the work that was going on through Wastewater Scan and through our partners at TEFI that allowed us to scale very fast. I will say we very quickly found, as the other groups have, that it was really dairy inputs. So like our personal experience, we monitored all these schools, and we thought it was very odd that we saw this late influenza peak at all of the schools that we're monitoring for. And we weren't doing an H5 specific assay, but it was like, why are we seeing so much influenza after the first peak in schools? And it turns out we actually retroactively went back and tested for H5, and it was positive. And we think it's the school milk input, because everybody gets school milk, and I think a lot of kids don't drink their milk, and it gets dumped down the drain. And that was that signal we're seeing. This also speaks to, and I'll stop talking about this, that like, what do we do with that information? And that's something that was really challenging. Okay, this is largely coming from dairy inputs. We can't really tell just using our standard assays whether or not this is reflective of human cases, right, or will be in the future. And so, like, what's the scale at which we should be monitoring this routinely? And I think that's something we're still discussing, but it has complicated even our school monitoring, because we want to report out at the schools that you have influenza in your school when it's just your milk, you know, that's positive for. Luckily, there's not that many infected herds anymore, so it's not positive anymore. But like, that's something we're very concerned about, and how we communicate that information. I don't know if anyone else wants to.
I'll say too that it really, our ability to be able to do this quickly, I think also demonstrated, and we knew this for a while, that wastewater was such a great like One Health approach for thinking about other contributing factors other than just human illness, right? But this particular example with H5 really gave us a lot of indication that we can look for other animal sources. We can be thinking about this from a more of a One Health framework. But it does, as Lauren said, complicate, you know, how we've typically interpreted trends over time with like SARS-CoV-2, for example. So I think we will have to like continue to be mobile and be able to stand up some of these subtyping or do sequencing to be able to confirm what we're seeing in wastewater, because it does have this potential. But of course, like with any benefit comes like the other side of the coin, right? Where you have to really think about how it's influencing your inference of what you're looking at over time. But the geospatial data is key to know what you're monitoring, what the inputs are.
If there's no other questions, I'll just add that like the ability to do that type of very quick work really differs by jurisdiction. Like, you know, we have New York, Utah, North Carolina. We are fairly large programs, like North Carolina, New York are now centers of excellence. If you talk to folks from other jurisdictions, their ability to do this type of lab work really differs, and there are some states that we've met with that have no bioinformatics capacity, and so they have to work with their academic partner in order to be able to do that. So just thinking, like, we might have really fast, really great data for North Carolina, but that won't tell us what's happening in some of these smaller jurisdictions, and so any type of plan really has to consider some of those lower resourced jurisdictions, I think, if you really want to get a good national picture of what's happening.
So I have lots of questions. I'm Christine Moe from Emory University, but I'll just start with one to be fair. So first of all, thanks to all the speakers, that was great. And thank you, Heather, for putting together such a great session. For application to low and middle income countries, you presented really elegant methods. What do you think would be the most useful and the most feasible to move into next for countries that are starting to do wastewater surveillance? Amy, Lauren, the other Amy.
This is kind of like a presidential debate where you don't actually have to answer the exact question, right? Yeah, I'll talk a little bit about that because I think it's a great question. And I think that a lot of the work that we're doing on kind of like the cusp of all of these new technologies that are being developed, it's really interesting to think about like where could we potentially have some leapfrogging for low income areas where they could benefit from this technology development. And I think like the first step is definitely thinking about just, PCR-based methods and the utility that they can provide in low resource settings. And what I see is like the biggest bottleneck in low income countries is it's not getting the capital equipment into place, it's getting the reagents on a regular basis imported into the country. And so I always wish that there was some international organization that could help with this, because I feel like it's literally the biggest constraint to being able to do more equitable global surveillance around the world. So that would be my first point. And then I think my second point is portable long-read sequencing, I think, also has a really important role to play. So we're seeing a lot of utility out of using nanopore sequencing to do tiled amplicon sequencing, but also thinking about some of these newer methods for targeted sequencing. I think that's also really valuable and is really exciting.
So just to add, I think that the most important point here is that we need this surveillance of low and middle income countries and rural areas, right? We can't sit here in high income countries and say, well, we got our surveillance. Because as we saw, the world's connected and it just takes some variant or strain or form of antibiotic resistance in some corner of the world, and there you go, right? There's a study with the traveling that was really cool. And so that means we need partnership. We can't just leave these folks off on their own. And Amy's right, I've seen the same thing, that you just can't get some of the reagents and supplies. But keep it simple, right? Extracting DNA isn't that hard, right? So if we could at least support them on site to collect samples, extract DNA, and then have some centralized facilities that do the sequencing or the qPCR or what have you. And a really nice thing about sequencing is that archival nature, that you can go back to it, and yeah, maybe you didn't catch that outbreak that time, but what can you learn from what happened so that we can start growing our capacity?
Can I piggyback off of that? I feel like there's also a lot of lessons that we can learn from kind of the data systems we need in place and the work to connect to remote places in the US, the non-sewered settings and inference to those areas are additional techniques we need. And even when we keep mentioning partnership, but I'm thinking about when MOHs are starting to think about this, is there a way to have that partnership with either our new centers of excellence or other groups that have thought through these different pieces? And I feel like part of this is not even, I would love to hear kind of thoughts from our health department partners of where there are areas, especially when you're thinking about jurisdictions that are just starting their own program. What are those pieces that might be more portable and could be applied in other settings, lower resource settings?
Well, I think that sample size question and tool is a huge piece. I think the question about sample size is huge. Because if you're resource constrained in any context, what's the minimum we have to do to get a representative sample? Where should we be sampling? Again, from the clinical side, there's a paper that came out from UNC that came with an Excel file that you could just plug in, this is what we want to do, these are our assumptions, and then it said, great, so we're gonna have to sequence 100 samples, and if I can just open an Excel file and do that very quickly, that's a very specific tool that doesn't require me to have any, I don't have a background in bioinformatics, I don't, so just really simple things like that, I think, personally, I love those. But I think that's also something that it might have to be adapted to different contexts, but we can, that can be shared widely.
Maybe I'll just build like very specifically about in the lab side. I think, a lot of labs don't have the ability to rapidly design assays, right? And so having assays be available, like sharing of sequences, there's a lot of interest in industry in this space, and they often will make these assays and sell them to health departments who are starting out, but these sequences are not publicly available for what they're using for primers and probes. And then to me, that kind of goes against this whole mission of trying to rapidly, expand it for public health. If you don't have, the sequences available, to even just like look at them and see how they compare to what other folks are using. And so I think that's something we need to be careful. I mean, we want to encourage like the people, industry to be excited about this, but we don't want to be at the expense of being able to rapidly expand.
Yeah, and kind of from the lab method side as well, I'll say the validation of some of those assays is, I think, where our biggest time sunk is spent from the domestic side, because we have partners that come out with these assays that are developed, and for us to implement them into routine surveillance, we need to validate them as part of our overall kind of process. And it takes a really long time for CDC to, validate an assay, for us to validate an assay at the state lab. But if we could reduce that time, then potentially those are available then for not just applicability within us in a health department, but when we're thinking about the global setting, right, as well, and how we can use those. And we in North Carolina have about 50% of our population is on private septic. So we are one of the few states that are, well below that national number. And some of what our work with UNC is really trying to focus on, especially with our Center of Excellence, is how we are looking at some of these smaller community septic systems and like smaller package treatment plants, and applying some of these methods for antimicrobial resistance, for some of these newer pathogens that we're thinking about. And if we can get those to kind of work within where we are in North Carolina, because we have a lot of rurality here, that to me has like a lot of applicability for some of these communities globally that we're thinking about trying to do some of this work for.
I can just, sorry, I know you guys may not be able to see our hands raised on the screen there. One of the things we're doing in New York here with our partners at Syracuse University is looking at human movement. And I think this applies to the rural communities and those that are not on sewered systems, because they're not stationary, right? People that live in rural areas are not stationary. They go to other places, they travel, they go on vacations, they commute, right? And so if we can understand those patterns and we can match those up with how either new diseases or emerging threats can move with humans, then we can better understand how those rural and low populated areas are, whether they need to be part of the network, or whether we have ways of understanding, how they can get exposed to certain agents. And maybe that is something that can be applied to other countries in other areas of low income, is understanding how people are moving from village to village, or to population centers to get food, or other aspects of their daily and annual movement. So yeah, I think that's a big part of it.
I was gonna hop in and just kind of a few things from our perspective. So we're heavily sewered. So of course we think about our non-sewered populations, but kind of when I'm thinking about rurality, I'm more thinking about the collaborations and work that I'm doing with the local health jurisdictions in those areas. And that can be quite challenging because, wastewater surveillance data is very new. And to us, it's not to us, but to them, to me going in there and being like, let's use this new data source for public health action. They're very resistant to implementing these new surveillance techniques because they simply just don't understand it, and they don't really understand like the public health actions that would occur from the findings that we have. And so I think that I think there's a few things that we need to kind of think about. I mean, one is like having clear public health actions. I loved what Ariel showed with her decision tree. Stuff like that, I think, is unbelievably helpful in getting those local health jurisdictions to kind of work with us, especially in those rural regions, having trainings and stuff like that. But more sustainable funding that shows that, that they should invest the time it takes to understand these surveillance systems. And I think this extends more than just in the United States, but like everywhere, that if we're able to sustain funding in places on the research side, on the academic, from academic institutions and from public health side, then people who are actually making the decisions in the public health actions and implementing it will actually spend the time it takes to be able to understand wastewater surveillance as a whole.
So, hey, thanks to the panel. This was all really super excellent. And I was wondering if I could ask people to comment on other kind of high-value targets that we haven't talked about today. We have, well, the conversation around vaccination in this country, is pretty fraught. And so I'm thinking about we should be using this for all vaccine-preventable diseases, probably. Planned Parenthood and other sexual and reproductive health services are going way down the tubes. Why aren't we looking at STIs? Thinking about arboviruses and other vector-borne diseases. So tell us about that. And I acknowledge that one of the huge challenges is validating assays before we need them. So, can we as a network, maybe this is a comment rather than a question, can we start thinking about those things and what this is going to look like down the road, because the public health conversation is going to move and change. And also because assays, we need these things for other places in the world beyond the United States where public health problems are very different, and the prevalent infections are also very different.
I can start by just talking about the vaccine, because that's exactly kind of the focus of Houston Center of Excellence has largely been using wastewater surveillance for vaccine preventable diseases, and then thinking about how we implement it in a way that actually encourages vaccinations in communities. And so I'll share just our success story, which if anyone's heard me speak before, I always talk about this, but we monitor at schools. And we now have a school alert program. So if we have a detection at the school, especially for things that are vaccine preventable, so like SARS-CoV-2, influenza, if you're a parent, you get a push alert. You have to subscribe to it. You don't just push alert. You get a push alert saying like we've detected this in your school, and if you have multiple, and it's information about what you can do, where you can get vaccinated. And if there's multiple detections at a school, they actually send a free vaccine clinic to that school. And what we've seen, and we've also started this for measles, mumps and rubella. Luckily, we haven't had any measles cases, but we've been doing this and expanding to other vaccine preventable diseases. And we've seen that offering these free, very convenient vaccine drives at these schools has been effective, not just for getting those vaccines up for the things that we're detecting, but a lot of times parents will just be like, oh, you can catch up on all your vaccines right now, right here. And so we'll see actually people get MMR vaccines just because they fell behind for their 4 or 5-year-old kid. And so we've seen it's been a great way to kind of encourage vaccines within the community. So I think it's a very important use case for wastewater surveillance. And the problem with facility level surveillance is that you're only getting that facility. So I think it's very effective for action, but it's limited in scope. So I think it's about how do we balance it so not only a few communities get access to this resource, because we're doing 50 schools, in a school district, that's hundreds and hundreds of schools. Why those 50 schools? Because they joined our program early on in the pandemic, but that's not really that. My point is there's a question of how do we expand this in a sustainable way.
I do think, there is the field of metagenomics and pathogen agnostic testing, and the whole goal there is that you don't have to have your select panel of targets, and you don't necessarily have to know what the next emerging thing is, that you could test for everything in one test tube. I don't know if you guys are the but it would be, and this is also, there's a part of human clinical diagnostics that's also trying to move towards like a metagenomic approach. there's a lot of pros, but I'll just say there's some very serious infrastructure and sort of data challenges that we would face on the public health side. And so we can't necessarily we can't do anything quickly, but it would take a very long time for us to be able to routinely do metagenomic sequencing. And so in the meantime, we really have to think about what's feasible and what's what we can pay for. And so when we think about what targets we're doing now and what our next set of targets are, I mean, Ariel can speak to this a lot better than me, but it really comes from conversations with our subject matter experts, guidance from groups like CDC. You have to be very, very judicious about what you choose because we just don't have the resources. But if we could test for everything by sequencing, that would be cool.
Dan has something. Dan?
Yeah, I'd just say for the vaccine derived or the vaccine preventable diseases, we also have to be careful about vaccine derived. So polio was our example in New York, right? That was not wild type, that was vaccine derived. VDP2 that was transmitted or being transmitted throughout the metropolitan area at the time as sequenced by CDC in our wastewater samples. And I believe measles also. We had measles cases in New York. We did some wastewater work on that. And it's hard to distinguish at the levels in wastewater whether that was vaccine signal or not vaccine signal. So that adds another twist to the vaccine preventable diseases side, is that we are vaccinating people with in some cases not had virus, right? So some of that's got to be factored in.
Do we have time for one more question? Yes.
Thanks for all the presentations. We heard a lot of great talks about innovative work around monitoring AMR in wastewater, and it seems like the news is very much focused on pathogen monitoring at this point. So I was curious if you could talk more about what are the use cases and applications, and where do you see the AMR monitoring going?
Yeah, I mean, I think this question kind of speaks to the fact that I almost feel like there's two frameworks that we need for wastewater surveillance. Like we have the emergencies and the outbreaks, and so we need to be able to respond quickly and roll out PCR-based and targeted sequencing for variant detection very quickly. But then I feel like there's a lot of things that we need to be tracking at the US level, but also at the global level. That's not like this respond really quickly. It's like we need to understand trends over time, and we need to develop these standardized methods to be able to understand what's happening over time so that we can be prepared and we can predict what's going to happen, in the future. And so I think that, the targeted metagenomics is a helpful direction because, it's hard when you're just looking for everything, then you don't know how to deal with that data. We really need to be thinking about how are we going to use the data that we're collecting, and how is that going to be used to look at historical trends. And so having this somewhere in between of not just these are all the ARGs that we found, and we can kind of look at quantification, but it's it's not super clear. We need to have a bit more targeted framework to really be looking at what we think is going to be important.
Yeah, and to build on that, I think AMR is an example where metagenomics, targeted or shotgun, is ideal, and exactly as you said that. I think there's some confusion sometimes that we did it for SARS-CoV-2 and it was about early warning, and we were focused on the epi, okay, how many people have this, and whereas with AMR right now, I think it's a slow pandemic, it's bigger picture, we need to see trends, we need to identify corners of the globe where there's a problem, and we can focus efforts. And I think that metagenomics in general is ideal for that. We can go back and look at emerging variants of genes. We can assemble data, look at bug drug combinations. one more thing, it's the pipelines, we do need user-friendly pipelines, because I'm hearing from a lot of people like, well, we get metagenomic data, now what? And that, shouldn't be the case. I mean, just like, I don't know how my computer works, but it works, right? And so I think we shouldn't be too far off from pipelines that, anybody can use.
I'll just project. I'll say,
We can also take them like out of the state.
yeah. I'll just say, I mean, I agree with all of that from the health department side, but I also think we're at this place where we don't quite have enough research yet to be able to think about implementation with AMRs into public health action. I don't think we have enough of those use cases yet in the research to really show. And I think some of the question marks around public health actionability come down to, can we look at trends over time, but also is this from a public health perspective more actionable at like the facility level, for example? What are the epi questions that still remain, which are like, what is the population truly at risk here? And who are the folks that we're trying to look at? And I think for some of those things, it's really different, right, for different groups. And it's also in the implementation, I think what's challenging for wastewater surveillance folks at a state level is that I have to coordinate with like 4 different groups. So whenever you're getting into like different pathogens, I'm going to an entirely different department within our communicable disease folks and having to train them about what is wastewater surveillance. The folks that respond and do public health action related to AMR or in facilities is a very different group than I work with for sequencing. So it's like having to make that use case across the whole health department, and CDC does this as well at the national level. It's really challenging because you have to be like you have to know just a little bit enough about everything to be dangerous, just dangerous enough to make the conversation interesting enough to hook them in. And you really have to think, and you have to know what evidence-based strategies are out there for some of these targets. And I think that applies for all the targets we've talked about here today. And how does wastewater actually fit into that picture? Is it like a clear value add? Like here's the missing trends we don't have in some of these areas? Or is it now I can help with cost allocation for you can use your clinical sequencing, right, a little bit more targeted than you can, because I can tell you with 10 wastewater samples a lot more than 10 clinical samples, right, about what variants are circulating in the state. So then you can use all of your resources to do a little bit more focused clinical target sequencing. So I think just like those conversations, and you have to like really think about how it fits into the overall public health response strategy that is evidence-based and currently, best practice.
Carrie?
So Ariel just brought up some stuff that like I think really pertains to CRS as well. Because so we've done some upstream sampling for CRS, and one of the things that like is really a problem for this, which probably seems really obvious to everyone here, but like, I don't know, I was a little bit surprised, was the resistance that we got from long-term care facilities. So we identified them as like, they're obviously the people that are at risk. And so when we're identifying these populations, we also have to know and handle the resistance from those communities that are actually going to do it because they were really not into it. They just didn't want to know, to be honest. And it drives me wild that that's where we are. But I mean, it certainly, that is, and that's something we're expecting. We're just starting up this program with CRS as one of our targets for all 35 of our facility level samples, but I'm keeping that in the back of my mind, how much resistance I'm going to get when I'm actually doing that. I think AMR is probably going to have similar issues.
Well, thank you so much. Thank you so much to our panelists. Really appreciate having this diverse group of folks here. We are at time, and thank you all for everyone coming in and joining this session.
We'll let you all go. Good to see you. Yeah, good to see you. Wish y'all were here. We'll see you in November at CDC. All right. I think I ended it, but I don't see it. All the problems, yeah. That's fine. Yeah.